So today I'm gonna maybe leaning a little bit more toward the graphical feature instead of the dynamical feature of reaction network. And specifically, I'm gonna talk about deficiency zero for random reaction networks under two random graph frameworks. The first one being Erdos Renyi, and the second one being Stochastic block model framework. So first of all, let me give you a very quick introduction of reaction networks and also the motivation for this project of mine. So biochemical reaction networks are very common in a lot of settings, from the microscopic ones like cell biology, protein, chemistry, to macroscopic ones, something like the study of epidemic disease or evolution biology, or population in ecology. So you can see it in a lot of other fields. So essentially, a reaction network model consists of two components. The first one is suggested by the name of the model. So we have a network or a graph component. And then the second component is a dynamics under such a graph. And usually there are two common choices for the dynamics. The first one being deterministic, ODEs, and the second one is stochastic. It is a continuous Markov chain. And not surprisingly, there is a strong connection between the two components in the sense that there are special network or graph structures, namely deficiency zero, endostaticity, and the special network or graph structures may ensure a lot of desirable behaviors of the dynamical systems. For example, existence and uniqueness of equilibrium point, or existence of stationary distribution, and a lot of stability properties. So thanks to a conversation between my advisor, David Anderson, and Robin Pimentel at UPenn, I came across some very interesting questions. And the questions are, so we know that there are some special structures that guarantee a lot of good features, good properties. Then how common or how prevalent these special structures are, and what a typical network with such features should look like. Is it sparse? Is it dense? How about the clusters? Do we have like a big cluster, or do we have a lot of smaller connected components? And in this talk today, I'm going to try to address these questions specifically for the structure deficiency zero. And in order to do that, I will present two random graph frameworks to generate random reaction networks. And then I'm going to study the scaling limit of the probability of deficiency zero as I send the number of species to infinity. So essentially, I'm studying the probability of deficiency zero for very, very large reaction networks. So just a very quick spoiler and maybe also a take-home message. So it turns out deficiency zero networks are often sparse. And in this talk, I'm going to quantify how exactly how sparse it should be to have deficiency zero with high probability. Okay, so that is a quick introduction. And here is the outline for the remaining of my talk. So first, I'm going to let you know about reaction network and give the definition on deficiency, and maybe give some intuition of what exactly is deficiency. And then I'm going to present two frameworks, two random graph frameworks for generating random reaction networks. The first one is Erdos Renyi, and the second one is Stochastic block model. And then in the next section, I'm going to use the random graph frameworks to study prevalence of deficiency zero. And finally, if time permits, I'm going to tell you maybe 2 or 3 future ideas and directions. Okay, so let's get started with reaction network. and the definition for deficiency. So here is a formal definition for reaction network. So a reaction network is fully specified if we know three sets: the set of species that interact with each other, the set of complexes, which are the linear combinations of species representing those used and produced in every reaction, and finally, we should have the set of reactions, right? So let me give you a quick example of a very popular model in epidemic disease, which is an SIR model. And let me open my note. So here is the SIR model. You can see the reaction graph, S plus I to 2I, S to I and to R. And usually there be also the rate constant over here, but it is not going to be important in this talk, so we can forget about that. So the reaction network can be fully specified from this reaction graph. And we can get the set of species from the reaction graph. Here we can see three species. So we have susceptible S, we have infected I, and we have recovered R. So we have three species, and we can also get the set of complexes from the reaction graph. So this is the first complexes, the second one, the third one, the fourth one, and then the last one. So I have 5 complexes: S plus I, 2I, S, I, and then R. And finally, we have 3 reactions. The first one is S plus I to 2I. The second one is S to I. And then the last one is I to R. So you can see either from the set of reactions or from the reaction graph, we can fully know a reaction network. Okay, so back to the slide. So this is another important representation of reaction network as an embedded graph. It is going to play a critical role later on, so I want to let you know right away. So let's say we have n species, then reaction network with n species can be embedded in Zn, and we can represent reactions by vectors. For example, here I have a reaction network with 2 species A and B, so I can embed it in Z2, and you can see here we have 3 reactions, and I can represent the 3 reactions by 3 vectors. The first one from 0, 0 to 3, 1. The next one from 3, 1 to 4, 4. And then the last one from 4, 4 to 0, 0. And I want to let you know that for the rest of the talk, I'm going to use reactions and the associated vectors interchangeably. So I hope that you will not get confused when sometimes I talk about reactions, but I may actually talk about the associated vectors. Right. So now we want to define deficiency, and before that, we need some important definitions. The first one is weak reversibility. This is a property that usually go together with deficiency 0, so I want to mention right away. So a network is weakly reversible if every connected component is strongly connected. So for example, if I have a reaction from A to A plus B, then there can be multiple reactions that take me back from A plus B all the way back to A. And another definition is related to the embedded graph representation. Remember that we can represent reactions by vectors. So we can talk about the linear subspace generated by all the vectors. And we call it the stoichiometric subspace of the network. We will denote the dimension by S. And from now on, whenever I mention dimension, that is just the dimension of the stoichiometric subspace. Right, so now we have everything we need for the definition of deficiency. So here's a definition. So the deficiency is equal to the number of complexes minus the number of connected component and then minus the dimension. So it may look a little bit mysterious at first, but I will try to make sense of this definition of deficiency. There's a lot of ways to make sense of deficiency, and this is just one way to understand it. So when we think about the name deficiency, it seems like something is missing, and then it turns out the thing that is missing here is going to be the number of potential dimensions that are missing due to linear dependence among reactions. So let me give you an example. So what I prepare here is not a reaction network yet. I just have a connected component structure. I have not put the actual complexes into this connected component structure, but just from this connected component structure, we can get the maximum number of dimension, right? So for the first connected component, we can have at most 3 minus 1 dimension, because we can have this one and this one if they are linearly independent, and the last one is going to be a linear combination of the first two, so we can have at most 3 minus 1 dimensions. And from the second connected component, we can have at most 2 minus 1 connected components. 2 minus 1 dimension. So the total number of maximum dimension is going to be the number of complexes minus the number of connected component. And in this case, we have maximum of 3 dimension. Okay, so now I'm going to try to put in some real complexes. Let's say I have empty set here, A here, and then B here. So the first connected component gives me exactly 2 dimension, because this one and this one are linearly independent. Now, here for the second connected component, if the reaction is 2A to 2B, then I do not gain an extra dimension just because this reaction vector is linearly dependent from this reaction vector. So it turns out I only have 2 dimensions. So the number of dimension is just 2, and you can see that the deficiency is the difference between the maximum possible dimension and the actual dimension, which is 1 in this case. That means I am missing 1 dimension because of the linear dependency between the 2 connected components. So that also means deficiency 0 implies some sort of linear independence among connected components. This notion of linear independence is going to be very important later on when we get to the proof of the prevalence of deficiency 0. Right, so that is the definition of deficiency. But why exactly is deficiency 0 an important concept in reaction network theory? It turns out that highly nonlinear system with deficiency 0 sort of mimic the behavior of a linear system. And in particular, deficiency 0 and weak reversibility imply a lot of good desirable dynamical properties. For example, in the deterministic model, we have existence and uniqueness of an equilibrium in every compatibility class, and we also have local stability. And in the stochastic model, we have existence of a very nice product form Poisson stationary distribution. And we have a lot more results related to deficiency 0. So it is a central concept in reaction network theory, and it is exactly why I want to study the prevalence of deficiency 0 among random reaction networks. Okay, so now after getting some understanding about reaction network and deficiency, I want to present two random graph frameworks for generating random reaction networks. So now if we think about it, if we want to get a framework for generating random reaction networks, we need to make some choices. We need to make the choice of vertices, and we need to make the choice of edge probability or edge wedge. So for the choice of vertices, what I'm going to choose is going to be all possible complexes in a class of reaction network called binary reaction networks. They are the reaction network which make up the majority of networks in biology and chemistry literature. And what exactly is a binary reaction network? So a reaction network is binary if every complex is either 0th order, which is an empty set. It can be unary, which is a single species, or it can be binary, which is where the sum of coefficients is exactly 2. So in particular, let's say we have n species, S1, S2, all the way to Sn. Then the set of vertices that I am going to consider is empty set, and then OSI, and then OSI plus Sj. Just a quick note here, I and j can be the same. In that case, we'll have something like 2Si. So now we get to the choice of edge probability, and the most basic choice, the most basic model is the Erdos Renyi model, which has uniform edge probability. So we can consider an Erdos Renyi random graph, Gn, where the set of vertices is empty set, OSI, OSI plus Sj. So we have an order of n square vertices. And we have Pn is the probability that there's an edge between any two vertices. And we can make a connection. We can create a reaction network from each random graph by throwing away all the isolated vertices. And then all the non-isolated vertices will become complexes, and all the edges will become reversible reactions. So here's one example. So when I have one species, then I have 3 possible complexes, empty set, A and 2A. And here's one realization when P is equal to 1/2. And I can get the associated reaction network, which is empty set to A reversibly, and then A to 2A reversibly. Now, if we have 2 species, then we'll have 6 possible complexes, empty set, A, B, 2A, 2B, and then A plus B. And here's one realization when P is 1/6. And we also have, we throw away A and 2A because they are isolated, and we have the associated reaction network with 2 pairs of reversible reactions from empty set to 2B, and then from B to A plus B. Right, so that is a basic Erdos Renyi framework. It is basic, it is straightforward, easy to understand, and not too difficult to work with. But you might think that the basic framework may not be realistic in all settings, and I totally agree with that. So after the first paper, we also have the second one, which utilize a more flexible framework that could generate random reaction networks in a lot of different settings. For example, let's just say I want to generate random reaction networks for open systems with a lot of inflow and outflow reactions, empty set to Si or empty set to Si plus Sj. Or maybe I just want to work with closed systems with very little or no inflow and outflow. Or maybe in some other setting, I just want to focus on the systems with certain conservation law. Let's say Si to Sj and just Si plus Sj to Sh plus Sk. So in order to generate random reaction networks with this underlying structure, what I can use is the stochastic block model, which essentially is just a generalized version of Erdos Renyi with weighted edge probability. So here is the detail of how to set up the stochastic block model. So recall that we have sort of like three types of vertices: the empty set, unary vertices, and then binary vertices. So we have like three blocks of vertices, and we can partition the set of edges according to the three blocks of vertices. For example, I'm going to let E01 to be the type of reactions from empty set to Si. So from empty set to unary vertices. And E11 is going to be the set of edges from unary to unary. E02 is going to be from empty set to binary, and then so on. So after partitioning the set of edges according to the types of vertices, we can introduce the control parameters alpha ij to control the edge probability in every type of reactions. We need a baseline, so we will choose the biggest group, E22, to have edge probability just Pn. But for other group, Eij, they will have edge probability n to the alpha ij, Pn. So for example, if I set alpha 01 and alpha 02 to be 0, then now you will see relatively little, or sometimes you will, with high probability, you will not see any inflow and outflow reactions. So if we set alpha 01 and alpha 02 to be 0, we can use it to model closed system with no inflow and outflow. And here is an example for an open system generated by this framework. So here, in particular, alpha 01 is 3, alpha 02 and alpha 11 is 2, and then alpha 12 is 1. The alpha are chosen so that you will see roughly the same amount of reactions in every type. And here I have 6 species, and I have the probability is 0.8 over n cubed. And here's one realization. You can see that we have a big cluster connected to empty set. They are the inflow and outflow reactions. And we have some smaller connected components that are not connected to the empty set. Okay, so now we have 2 frameworks for generating random reaction networks, and we can study the prevalence of deficiency 0 under the 2 random graph frameworks. So the first framework is going to be Erdos Renyi, and it's going to be the easier framework to work with. So in order to quantify the prevalence of deficiency 0, we are establishing what is called a threshold function for deficiency 0. And we found the threshold function to be 1 over n cubed. And this is the main result. So when Pn is sufficiently large, when it is asymptotically larger than 1 over n cubed, then we have deficiency 0 with very low probability. So when the graph is dense enough, then it is very unlikely we will have deficiency 0. And when Pn is sufficiently small, when it is asymptotically smaller than 1 over n cubed, then we have deficiency 0 with high probability. So when the graph is sparse enough, then we will likely to have deficiency 0. So let's take a look at an example to understand the statement a little bit more. So let's say I have 10 species, and we have the set of vertices, empty set, O the Si plus Sj. So the number of vertices is roughly n squared over 2, so roughly 50 vertices in this case. And the number of possible edges is going to be v choose 2, so roughly n to the 4th divided by 8. And in this case, it is roughly 1,200 possible edges. So what happens if Pn is much bigger than 1 over n cubed, which is 1 over 1,000? Let's say if Pn is 1 over 10, then the expected number of edges is going to be this Pn multiplied with the number of possible edges. So 1 over 10 times 1,200. So we have like more than 100 edges. And if we think about it, if we have a lot of edges, then the first term, this term, the number of complexes minus the number of connected components is going to be a big number. So let's say 100 plus. However, the dimension S is bounded by the number of species. So S can be at most 10. And you can see that the deficiency is going to be most likely positive, right? Because this is a very big number, and then S is going to be small. So deficiency is going to be most likely positive. However, if Pn is quite small, if Pn is just like, let's say 1 over 2,000, then the expected number of edges is going to be 1 over 2,000 multiplied with this total number of possible edges, and we have something like 1/2. So we may see maybe 0, 1, or 2 edges. Likely. And we have 10 dimensions, so if we only see 0, 1, or 2 edges, then it is likely that they are linearly independent. And because of that, we will have deficiency 0. So that is only for n equal to 10. And here, when we set n to infinity, you can imagine that when Pn is bigger than the threshold, then the probability of deficiency 0 will go to 0. And when Pn is much smaller than the threshold, then the probability of deficiency 0 will go to 1. So to recap, this means a random reaction network needs to be sparse enough to have deficiency 0 with high probability. And I quantify how sparse it should be, which is Pn should be much smaller than the threshold 1 over n cubed. So now we can talk a little bit about the proof. So the easier direction is when Pn is sufficiently large, when Pn is much larger than 1 over n cubed. We will show that the probability of deficiency 0 will be very small and will tend to 0. And the intuition is similar to the example that I just showed you before. There are likely just too many complexes or non-isolated vertices, and that makes the deficiency strictly positive. In particular, for the outline of the proof, first we can show the upper bound, a particular upper bound for the number of complexes. And it turns out that the number of complexes or non-isolated vertices has to be less than or equal to 2n. This is going to be a quick workout. So let me quickly show it to you. It's just going to be 2 lines. So we go back to the definition of deficiency, which is C minus L minus S. And if we have deficiency 0, then the side of C will be equal to L plus S. And because every connected component has at least 2 complexes, so the number of connected components is less than C over 2. And then remember that the dimension is bounded by the number species n. So we have this. And we'll have C over 2 is less than or equal to n. And we have a very easy upper bound for C, which is 2n. Okay, so we have an upper bound for the number of complexes. And when Pn is asymptotically bigger than 1 over n cubed, I can show that with high probability, the number of non-isolated vertices will exceed the bound. And in fact, in the proof, this is done by investigating the number of isolated vertices instead. It is actually easier to work with the number of isolated vertices. And finally, if we are careful with the calculations and inequalities, then we can get like a slightly better result. With Pn just need to be slightly larger than n over n cubed instead of asymptotically bigger than 1 over n cubed. Okay, so that is an easy direction. The more difficult direction is when Pn is sufficiently small, when Pn is asymptotically smaller than 1 over n cubed. We will show that with high probability, the network will have deficiency 0. So here are some quick initial analysis from random graph theory. So when Pn is much smaller than 1 over n cubed, with high probability, we will see that the graph will contain only connected component of size at most 2. So we'll see a lot of small connected component and then a lot of isolated vertices. And also, we will most likely see the reaction type Si plus Sj to Sh plus Sk. So binary to binary, just because the overwhelmingly large amount of this type of reactions compared to the other type. Now, recall from earlier that the concept of deficiency 0 is closely related to linear, sort of like linear independence among the connected components. So the remaining of the proof eventually boil down to a discrete random matrix questions. So suppose that a matrix has n rows, right? Because we have n species, and the columns have some specific structure. Recall from earlier that the most dominant type of reaction that we mostly see is this binary to binary. So the columns will have a very specific structure. 2 random entries will be 1. Another 2 random entries will be -1. And then the other entries will be 0. And the question becomes, how many columns can the matrix have until linear independence is broken? And proving this is not easy. This is a highly non-trivial question, a lot of technical details. But if you are curious about the key idea of the proof, then what I did is I enumerate on the minimally dependent set of columns. And by minimally dependent, I mean the set, if you take away any element, you will get an independent set. So any proper subset is independent, basically. So I enumerate on minimally dependent set, and then I analyze the number of zero rows. And after that, it's just a lot of technical details with a lot of inequalities and combinatorics. So I'm not going to mention the whole detail of the proof here. So that is prevalence of deficiency zero under Erdos Renyi framework. And the next framework, stochastic block model framework, is much more challenging. But essentially, we will also be able to find a threshold functions. So remember that in the stochastic block model framework, we have the control parameters alpha I, j to control the edge probability of different type of edges. So given any choice of alpha I, j, we can actually find the threshold function Rn and then alpha I, j algorithmically using a set of conditions for deficiency zero. So I will have the set of conditions in the next slide. But before that, I think it's going to be better if we get some intuition of the set of conditions. So here, instead of listing the conditions, I'm going to list the situations where we see a random network with positive deficiency. So the situation where we can break deficiency zero. So the first situation is if we have too many reactions in E02, E12, and E22. The reason is if we have too many reactions of these types, then we also have too many complexes. And now it loops back to the argument in the Erdos Renyi case. If we have too many complexes, then the network will have deficiency zero, positive deficiency with high probability. The second situation where we can break deficiency zero is if we have too many linear reactions. So too many reactions in E11. If we have too many reactions in E11, we will almost exhaust the possible dimensions. And if we add some more reactions, and precisely we only need two more reactions from E02, E12, or E22, then with high probability, we can break linear independence. And because of that, we will not have deficiency zero. And the last situation is when we have abundant inflow and outflow, let's say for open system, and we have enough of other reactions, we can also break linear independence, and we will not have deficiency zero. And it turns out that if we are not in any of the situation above, we actually have deficiency zero with high probability. So this is the precise statement of the theorem. It looks very technical, so I will not try to explain this in more detail, but I do want to mention that the proof of the difficult direction also leads to a similar discrete random matrix problem, but in this case, the discrete random matrix problem is much more challenging because now we have a lot of different types of columns. We can have 1, 1, minus 1, minus 1. We can have 1, 1, minus 1. We can have 1, minus 1, and we also have some canonical vectors among the columns. There's also some constraints between the different types of reaction, and because of that, the proof becomes much more difficult and much more technical. Okay, so that is some intuition and some result on the stochastic block model framework. Let me just quickly tell you maybe two future ideas that I can develop from this project. So going from the Erdos Renyi model to the stochastic block model, that is motivated by the fact that I want to make the random graph model more realistic. And another way to make the random graph model more realistic is to consider the active levels of species. For example, in protein interaction network, some protein may be active and interact with a lot of other protein, while some protein can be inactive and do not have a lot of connections. So we can incorporate the activeness of species by a random vertices and random edges framework. We can assign different weights, Wi, for different species, Si, based on their activeness. And we can let the vertex Si plus Sj to appear with probability proportional to the product of the weights. And another direction that I want to work on is instead of studying the prevalence of deficiency 0, we can also use the random graph frameworks to study the prevalence of other structures. For example, we have endostaticity and asynchronicity, which gained interest recently in the theory of reaction network. This is going to be very challenging and also very interesting because there's no simple characterization of endostaticity, for example. And I also know from some people in biology that they are interested in learning about the frequency or the prevalence of autocatalytic cycles in random reaction networks. Okay, so that are two future directions. And here is just a very quick take-home message for the talk. So in the two projects, we designed two frameworks to generate random reaction networks. And we can use the two frameworks to study prevalence of network structures, such as deficiency 0. And in particular for deficiency 0, the random networks have to be sparse, so low edge probability. And we quantify how sparse the random networks have to be by using threshold function. And I think that is all I have for the talk today. Thank you very much for listening, and please let me know if you have any questions.